Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Robert Remak stands as a pivotal, though often underrecognized, figure in the history of 19th century science. His meticulous investigations into the fundamental building blocks of life and the intricate architecture of the nervous system yielded insights that irrevocably shaped the trajectory of both cell biology and neuroanatomy. Remak's work addressed core questions that preoccupy the scientific community: how do cells arise, and how is the nervous system organized? These were not abstract philosophical musings, but concrete empirical problems demanding solutions grounded in careful observation and rigorous experimentation. Prior to Remak's interventions, the prevailing understanding of cell formation, largely attributed to Matthias Sladen and Theodor Schwann, posited a process of free cell formation, or cytoplasm, or asterisk. This theory suggested that new cells arose from an undifferentiated organic substance, a notion akin to spontaneous generation at the microscopic level. Such a concept, while seemingly explanatory at the time, lacked empirical support and failed to account for the dynamic processes observed during tissue development. It was within this intellectual climate that Remak began his groundbreaking research. Remak's approach was characterized by an unwavering commitment to direct observation. Employing advanced microscopic techniques and meticulous histological preparations, he scrutinized cellular processes with unparalleled precision. His investigations, particularly those focused on developing tissues, revealed a strikingly different picture of cell genesis. Instead of arising de novo from amorphous matter, Remak observed that cells invariably originated from pre-existing cells through a process of division. This revolutionary conclusion, encapsulated in the now famous dictum Omnis cellular, every cell arises from a cell, challenged the very foundation of Sladen and Schwann's theory. The impact of this seemingly simple statement was profound. It established a clear line of cellular descent, linking each new cell to its progenitors and providing a crucial framework for understanding growth, development, and inheritance. Remak's meticulous observations provided the empirical bedrock for this principle, demonstrating through detailed microscopic studies the process of cell division and the continuity of cellular material. Remak's 150 publication, titled Concerning the Origin of Blood Cells and a Third Form of Blood Cell in General, provides a tangible example of his meticulous approach. In this work, he detailed his observations of blood cell formation, providing compelling evidence that new blood cells arose through the division of pre-existing cells. This was a direct challenge to the cytoplasm theory, which held that blood cells, like other cells, arose from undifferentiated organic matter. Remak's detailed descriptions and illustrations provided a clear alternative, demonstrating the power of microscopic observation to unravel fundamental biological processes. The modern relevance of Remak's contribution to cell theory is undeniable. Cell theory remains a cornerstone of modern biology, underpinning our understanding of everything from development and disease to evolution and inheritance. The principle that all cells arise from pre-existing cells is a foundational concept taught in introductory biology courses worldwide. While the mechanisms of cell division have been elucidated in exquisite detail since Remak's time, the fundamental principle he established remains unchanged. Without this understanding, modern fields like genetics, molecular biology, and cancer research would be unthinkable. Remak's insights into cell division did not emerge in a vacuum. He drew inspiration from the work of earlier embryologists, most notably Carl Ernst von Baer. Von Baer's meticulous studies of vertebrate development, particularly his discovery of the mammalian ovum, emphasized the importance of careful observation in understanding biological processes. Von Baer's emphasis on epigenetic development, the idea that development proceeds through a series of gradual transformations rather than preformation, resonated with Remak's own observations of cell division. Remak extended von Baer's emphasis on observation to the cellular level, revealing the dynamic processes that underlie embryonic development. He saw the cell as a fundamental unit of organization, analogous to the individual organism in von Baer's embryological framework. The influence of Remak's work extended to numerous scientists, most notably Rudolf Virchow. While Virchow is often credited with popularizing the cell theory, Remak's contributions were initially downplayed. Virchow's famous dictum, Omnis cellular is a direct appropriation of Remak's earlier formulation. While Virchow played a critical role in disseminating and promoting this concept within the medical community, the intellectual debt to Remak is undeniable. The unfortunate initial lack of proper attribution highlights the complex dynamics of scientific discovery and the challenges faced by those whose contributions are not immediately recognized. Virchow's adoption of Remak's principle, even with delayed acknowledgement, cemented its place in the scientific canon and influenced generations of researchers. Beyond his contributions to cell theory, Remak also made significant advances in neuroanatomy. At the time, the structure and organization of the nervous system remained largely mysterious. While the existence of nerve fibers had been established, their precise origins and connections were poorly understood. One particularly perplexing question concerned the nature of unmyelinated nerve fibers, delicate structures that lacked the insulating myelin sheath characteristic of many other nerve fibers. Remak's meticulous anatomical investigations led him to the groundbreaking discovery that these unmyelinated nerve fibers, which he termed Remak fibers, originated in the brain. This discovery challenged the prevailing view that all nerve fibers were essentially uniform in structure and origin. By tracing the course of these fibers, Remak demonstrated their connection to the central nervous system, providing crucial insights into the organization of the peripheral nervous system and its relationship to the brain. This finding had profound implications for understanding the function of the nervous system. It suggested that different types of nerve fibers might serve distinct roles in transmitting signals and coordinating bodily functions. Remak's identification of Remak fibers opened new avenues of research into the diversity and complexity of the nervous system, paving the way for future discoveries about the specific functions of different nerve fiber types. Remak's contributions to neuroanatomy, Like his work on cell division, were rooted in careful observation and meticulous experimentation. He employed advanced histological techniques to visualize nerve fibers and trace their connections, providing a detailed anatomical map of the nervous system. His work laid the foundation for future studies of neural circuitry and the mechanisms of nerve impulse transmission. He meticulously documented the structure of ganglia and their connections to both myelinated and unmyelinated nerve fibers. The influence of Remak's neuroanatomical work can be seen in the research of later neuroscientists who built upon his findings to develop a more complete understanding of the nervous system. His meticulous descriptions of nerve fiber structure and organization provided a crucial foundation for subsequent investigations into the cellular and molecular mechanisms of neural function. He demonstrated that nerve fibers were not simply passive conduits, but complex structures with distinct properties and functions. The meticulousness that characterized Remak's investigations into nerve fibers extended to his equally significant contributions to embryology. Understanding the earliest stages of vertebrate development posed a considerable challenge to 19th century biologists. Remak tackled this problem through painstaking dissection and microscopic examination of developing embryos, particularly those of the chick. His dedication to detailed observation yielded a groundbreaking understanding of germ layers, the fundamental tissues from which all organs and structures of the vertebrate body arise. Christian Pander's earlier research provided a conceptual springboard. Pander identified 3 primary layers in developing chick embryos, yet his descriptions lacked the precision and comprehensive detail that would become Remak's hallmark. Remak built upon Pander's foundation, meticulously characterizing the formation, differentiation, and ultimate fate of these layers. He refined Pander's initial observations, providing a far more granular understanding of their composition and developmental potential. Remak's work culminated in the definitive identification and description of the 3 germ layers: the ectoderm, the mesoderm, and the endoderm. The ectoderm, the outermost layer, gives rise to the epidermis, the nervous system, and various sensory organs. The mesoderm, the middle layer, differentiates into muscle, bone, cartilage, blood, and the urogenital system. The endoderm, the innermost layer, forms the lining of the digestive tract, the respiratory system, and associated organs such as the liver and pancreas. His 1855 publication, Untersuchungen über die Entwicklung der Wirbeltiere, Investigations on the Development of Vertebrates, stands as a landmark in the history of embryology. Within its pages, Remak systematically detailed the formation and subsequent development of each germ layer. He traced their cellular origins, described their histological characteristics, and meticulously documented their differentiation into specific tissues and organs. The publication provided a crucial framework for understanding the complex processes of organogenesis, the development of organs. This framework established a new paradigm for studying embryonic development. Remak's meticulous observations offered a clear roadmap for understanding how a relatively simple ball of cells transforms into a complex, multilayered organism. He elucidated the precise sequence of events through which each germ layer gives rise to its specific set of tissues and organs. The clarity and precision of his descriptions set a new standard for embryological research. The concept of germ layers, as defined and elaborated by Remak, quickly became a cornerstone of developmental biology. It provided a unifying principle for understanding the development of all vertebrate animals. Subsequent generations of embryologists built upon Remak's foundation, using the germ layer concept as a framework for investigating the molecular and cellular mechanisms that govern embryonic development. Wilhelm Sr., a prominent Swiss anatomist and embryologist, stands as a prime example of someone directly influenced by Remak's germ layer theory. His SR adopted and further refined Remak's concepts, incorporating them into his own influential work on human embryology. He expanded upon Remak's descriptions, providing even greater detail regarding the cellular and histological characteristics of each germ layer. His SR also developed a more sophisticated understanding of the signaling pathways that regulate germ layer differentiation. The enduring relevance of germ layer theory speaks to the profound impact of Remak's work. It remains a fundamental concept in developmental biology textbooks and curricula worldwide. Students of biology continue to learn about the ectoderm, mesoderm, and endoderm as the foundational tissues from which all vertebrate organs and structures arise. The principles Remak elucidated continue to inform research into developmental disorders, regenerative medicine, and the evolution of vertebrate body plans. His meticulous approach to embryological research, characterized by careful dissection, detailed microscopic observation, and systematic documentation, served as a model for future generations of scientists. He demonstrated the power of combining anatomical observation with histological analysis to unravel the complexities of embryonic development. His commitment to precision and detail transformed the field of embryology, elevating it from a descriptive science to an analytical discipline. The legacy of Remak's embryological investigations extends far beyond the confines of the 19th century. His discovery and detailed characterization of the germ layers provided a crucial foundation for understanding the fundamental processes of vertebrate development. His work continues to inform and inspire researchers in developmental biology, regenerative medicine, and evolutionary biology. The germ layer theory, a testament to his intellectual rigor and meticulous observation, remains a cornerstone of modern biological thought. The meticulousness that defined Remak's embryological pursuits also permeated his forays into the realm of electrotherapy, a burgeoning field grappling with the complexities of neurological disorders. 19th century medicine faced a significant challenge in effectively addressing ailments affecting the nervous system. Conditions such as neuralgia, paralysis, and various forms of nervous debility presented a therapeutic puzzle, spurring investigation into novel treatment modalities. Remak envisioned a solution rooted in the precise application of electrical currents to targeted areas of the body. This approach stemmed from his deep understanding of nerve function and his belief that electricity could be harnessed to stimulate, modulate, and ultimately restore impaired neural pathways. He hypothesized that specific electrical parameters, carefully calibrated and delivered, could alleviate pain, improve motor function, and promote overall neurological well-being. His publications in clinical practice provide a detailed account of his electrotherapeutic techniques. Remak meticulously documented the application of electrical currents for conditions like neuralgia characterized by intense nerve pain. He advocated for the use of specific waveforms and frequencies tailored to the individual patient and the specific nature of their ailment. In cases of paralysis, Remak employed electrotherapy to stimulate muscle contraction and prevent atrophy, aiming to restore voluntary movement. He believed that electrical stimulation could re-educate the nervous system, prompting it to re-establish connections and regain control over paralyzed limbs. One can find instances of Remak's influence in the medical literature of the time. Physicians grappling with the treatment of intractable pain often turned to electrotherapy as a potential avenue for relief. While the field ultimately faced challenges due to inconsistent results and the rise of alternative therapies, Remak's contributions helped to establish electrotherapy as a legitimate, albeit controversial, treatment option. Michael Faraday's groundbreaking discoveries in electromagnetism provided the foundational scientific principles upon which Remak's electrotherapeutic work rested. Faraday's experiments demonstrated the intimate relationship between electricity and magnetism, paving the way for the development of electrical devices and technologies. Remak drew upon Faraday's insights to understand how electrical currents could interact with biological tissues, influencing nerve function and muscle activity. Furthermore, Dechen de Beauvoin's pioneering work on electrodiagnosis directly shaped Remak's approach to electrotherapy. Dechen, a French neurologist, utilized electrical stimulation to map the facial muscles and diagnose neurological disorders. His meticulous observations and detailed anatomical knowledge inspired Remak to adopt a similarly rigorous and systematic approach to electrotherapeutic interventions. Dechen's influence is evident in Remak's emphasis on precise electrode placement and careful monitoring of patient responses. The legacy of Remak's electrotherapeutic endeavors is complex. Correct electrotherapy, as practiced by Remak, has largely fallen out of favor in modern medicine. The field suffered from a lack of standardization, inconsistent results, and the emergence of therapies with superior efficacy. Moreover, the misuse of electrotherapy by unscrupulous practitioners tarnished its reputation, contributing to its decline. However, Remak's work laid the groundwork for later developments in neurostimulation techniques. Modern approaches, such as transcutaneous electrical nerve stimulation, or TENS, and deep brain stimulation, owe a debt to the early pioneers of electrotherapy. These techniques, refined and validated through rigorous scientific investigation, continue to play a role in the treatment of chronic pain, movement disorders, and other neurological conditions. TENS, for example, utilizes low-intensity electrical currents to stimulate sensory nerves, thereby blocking pain signals from reaching the brain. Deep brain stimulation involves the implantation of electrodes in specific brain regions, delivering electrical impulses that modulate neural activity and alleviate symptoms of Parkinson's disease, essential tremor, and other neurological disorders. These modern techniques, while far removed from the crude electrotherapeutic devices of the 19th century, share a common ancestry with Remak's pioneering efforts. The application of electrical stimulation to modulate neural function continues to be an area of active research and development. Scientists are exploring the potential of electrotherapy to treat a wide range of conditions, including depression, anxiety, and even Alzheimer's disease. While the challenges are significant, the prospect of harnessing the power of electricity to restore neurological health remains a compelling goal. Remak's contributions to electrotherapy, viewed through the lens of history, offer a valuable lesson about the evolution of medical knowledge. His work reflects the early attempts to understand and manipulate the nervous system using electrical currents. While his specific techniques may have been superseded by more advanced approaches, his pioneering spirit and his commitment to scientific rigor paved the way for future advancements in neurostimulation. Moreover, Remak's electrotherapeutic work underscores the importance of ethical considerations in medical practice. The potential for misuse and abuse inherent in electrotherapy highlights the need for careful regulation, informed consent, and a commitment to patient well-being. The history of electrotherapy serves as a cautionary tale, reminding us of the ethical responsibilities that accompany the application of any medical technology. The field of neurostimulation continues to evolve, driven by advances in neuroscience, engineering, and materials science. Researchers are developing more sophisticated devices and techniques that allow for precise targeting of specific brain circuits. The ability to modulate neural activity with greater precision holds the promise of treating a wide range of neurological and psychiatric disorders with minimal side effects. Remak's early work in electrotherapy, while imperfect, represents a crucial step in the long journey toward understanding and treating diseases of the nervous system. His contributions, viewed in their historical context, provide valuable insights into the evolution of medical knowledge and the ongoing quest to improve human health. The legacy of his electrotherapeutic endeavors, therefore, is not defined by the specific techniques he employed, but rather by the enduring inspiration he provided to those who sought to unlock the secrets of the nervous system and harness the power of electricity for therapeutic purposes. The exploration of physiological processes through a mathematical lens represents another facet of Robert Remak's multifaceted scientific career. While his contributions to cell theory and neuroanatomy are more widely celebrated, his forays into mathematical biology, specifically his work related to diabetes, offer a glimpse into his forward-thinking approach to understanding disease. The challenge at the time was to move beyond purely descriptive accounts of metabolic disorders and instead develop quantitative methods for analyzing and predicting their behavior. Remak sought to apply mathematical principles to the study of diabetes, aiming to identify patterns in patient data and construct models that could shed light on the underlying mechanisms of the disease. His efforts, though less prominent than his other discoveries, demonstrate an early appreciation for the potential of mathematical modeling in physiology. He published papers exploring the application of mathematical analysis to the study of diabetes. These works, while perhaps lacking the sophistication of modern bioinformatics tools, laid a foundation for future researchers interested in quantifying biological processes. The specific equations or models Remak developed in relation to diabetes may not be in widespread use today. The enduring significance of his work lies in its pioneering spirit. He advocated for the integration of quantitative methods into medical research. This approach foreshadowed the rise of modern bioinformatics and systems biology, fields that rely heavily on mathematical models to understand complex biological systems. Early researchers applying statistical methods to medical data provided a direct influence on Remak's approach. These pioneers, often working working with limited data sets and computational tools, demonstrated the potential of quantitative analysis in identifying correlations and trends in patient populations. Remak, building on their foundation, sought to extend these methods to the study of specific diseases such as diabetes, with the goal of developing predictive models. Subsequent researchers who developed more sophisticated mathematical models of physiological systems were directly influenced by Remak's early attempts. These scientists, armed with more powerful computational tools and a deeper understanding of biological processes, were able to create increasingly complex and accurate models of human physiology. The work of figures like Dennis Noble, whose work on the first detailed computer model of the heart exemplifies this progression, finds an early echo in Remak's ambition to quantify the intricacies of disease. The modern relevance of Remak's early attempts to model diabetes mathematically is undeniable. Today, bioinformatics and systems biology are essential tools for understanding and treating a wide range of diseases. Researchers use mathematical models to analyze genomic data, predict drug responses, and design personalized treatment strategies. Remak's work, though rudimentary by today's standards, represents a crucial step in the evolution of these fields. Consider, for example, the development of algorithms for predicting blood glucose levels in patients with diabetes. These algorithms, which are used in continuous glucose monitors and insulin pumps, rely on mathematical models to forecast how blood sugar will respond to food intake, exercise, and insulin injections. Remak's early attempts to model diabetes, though less sophisticated, share the same goal to use mathematics to understand and predict the behavior of a complex physiological system. Another example can be found in the field of drug discovery. Pharmaceutical companies increasingly rely on mathematical models to simulate the effects of drugs on the human body. These models, which are based on a deep understanding of molecular interactions and physiological processes, can help researchers identify promising drug candidates and optimize treatment regimens. Remak's work, while focused on a specific disease, highlights the broader potential of mathematical modeling in medicine. The historical context of Remak's work is also important to consider. In the mid-9th century, medicine was still largely based on observation and intuition. The use of quantitative methods was relatively uncommon, and many physicians were skeptical of the value of mathematics in understanding disease. Remak, therefore, faced an uphill battle in advocating for the integration of mathematical principles into medical research. He persisted in his efforts, publishing papers and presenting his findings at scientific conferences. Although his work did not immediately revolutionize the field of medicine, it helped to pave the way for future generations of researchers who would embrace the power of mathematical modeling. His work serves as a reminder that scientific progress often occurs incrementally, with each generation building on the foundations laid by those who came before. Remak's contributions to mathematical biology, though less widely known than his other discoveries, demonstrate his intellectual curiosity and his commitment to using the tools of science to understand the complexities of human health. He recognized the potential of mathematics to transform medicine, and he dedicated his career to advancing this vision. His work serves as an inspiration to researchers who continue to explore the intersection of mathematics and biology. The influence of figures like Carl Ludwig, who championed a mechanistic and quantitative approach to physiology, likely played a role in shaping Remak's interest in mathematical biology. Ludwig's work, which emphasized the importance of physical and chemical principles in understanding biological processes, provided a framework for Remak's own investigations. Ludwig's contributions to hemodynamics, for example, demonstrate the power of applying mathematical analysis to the study of physiological systems. Furthermore, Remak's work on diabetes can be seen as a precursor to the development of systems biology, a field that seeks to understand biological systems as integrated networks of interacting components. Systems biologists use mathematical models to analyze the complex relationships between genes, proteins, and metabolites, with the goal of understanding how these interactions give rise to emergent properties at the level of the whole organism. Remak's early attempts to model diabetes, though less comprehensive, share the same underlying goal to understand how the different components of a physiological system interact to produce a specific disease state. Consider, for example, the modern use of network analysis in studying diabetes. Researchers use network models to identify key genes and proteins that are involved in the development of the disease. These models can help to identify potential drug targets and to predict how different treatments will affect the overall system. Remak's work, while lacking the sophistication of modern network analysis techniques, represents an early attempt to apply a systems-level approach to the study of diabetes. The limitations of Remak's mathematical work should also be acknowledged. He lacked access to the sophisticated computational tools and large datasets that are available to modern researchers. His models were likely based on relatively simple equations and limited data, which would have restricted their accuracy and predictive power. Despite these limitations, his work represents a valuable contribution to the history of mathematical biology. The enduring legacy of Remak's mathematical contributions, therefore, is not defined by the specific models he developed, but rather by the enduring inspiration he provided to those who sought to quantify the complexities of human physiology. His work serves as a reminder that scientific progress often requires a willingness to embrace new approaches and to challenge existing paradigms. He paved the way for future generations of researchers who would harness the power of mathematics to understand and treat disease. Robert Remak's scientific journey, a testament to meticulous observation and innovative thinking, left an indelible mark on the landscape of modern biology and medicine. His contributions, spanning cell theory, embryology, neuroanatomy, electrotherapy, and early mathematical biology, demonstrate a remarkable interconnectedness, each facet illuminating the others. His work, however, unfolded against a backdrop of societal prejudice and institutional barriers, a reality that significantly impacted the recognition he deserved during his lifetime. Remak's meticulous work on cell division, culminating in his assertion that cells arise from pre-existing cells, omnicellular acellular stands as a cornerstone of modern cell theory. This principle, challenging the prevailing notion of spontaneous generation, provided a fundamental framework for understanding growth, development, and disease. The meticulous observations he made using the limited microscopic technology available at the time laid the groundwork for future advances in genetics and molecular biology. His influence reverberates through the work of scientists who built upon his foundation, including Walt Fleming, who subsequently described the process of chromosome separation during cell division, a phenomenon Remak had keenly observed in its nascent stages. The implications of this work extend to contemporary cancer research, where understanding the mechanisms of uncontrolled cell division is paramount. In embryology, Remak's detailed studies of germ layer formation revolutionized the understanding of early development. He identified the three fundamental layers, the ectoderm, mesoderm, and endoderm, and traced their origins to specific tissues and organs. This discovery provided a crucial roadmap for understanding how complex organisms develop from a single fertilized egg. His work directly influenced later embryologists such as Hans Spemann, who, through his Nobel Prize-winning work on embryonic induction, further elucidated the mechanisms by which cells communicate and interact during development. The insights gained from Remak's embryological studies continue to inform our understanding of developmental disorders and birth defects. Remak's pioneering investigations into neuroanatomy yielded significant insights into the structure and function of the nervous system. He identified nerve fibers as extensions of nerve cells, a concept that challenged the then prevalent view of the nervous system as a network of interconnected tubes. His meticulous dissections and microscopic observations provided crucial evidence for the neuron doctrine, the now universally accepted principle that the nervous system is composed of discrete cells called neurons. Virchow, a pioneer of modern neuroscience, acknowledged Remak's foundational work in establishing a cellular basis of the nervous system. Remak's anatomical discoveries underpin much of contemporary research into neurological disorders, including Alzheimer's disease and Parkinson's disease. His exploration of electrotherapy, while controversial in its time, reflected a genuine attempt to understand and harness the therapeutic potential of electricity. He meticulously investigated the effects of electrical stimulation on nerves and muscles, seeking to alleviate pain and restore function in patients with neurological disorders. While some of his methods might appear rudimentary by modern standards, his commitment to careful observation and rigorous experimentation laid the groundwork for future advances in electrophysiological therapies, such as transcranial magnetic stimulation and deep brain stimulation. The ethical considerations surrounding electrotherapy, particularly the potential for misuse, remain relevant today, underscoring the importance of responsible innovation and patient-centered care. Remak's early forays into mathematical biology, though limited by the available tools and data, demonstrated a visionary approach to understanding physiological processes. His attempt to quantify the complexities of human physiology, including his work on diabetes, foreshadowed the development of modern systems, biology, and computational medicine. His legacy in this area resides less in specific models and more in the enduring inspiration he provided to those who sought to apply mathematical principles to biological problems. Researchers continue to develop sophisticated mathematical models to understand and predict disease progression, building upon the foundational principles established by pioneers like Remak. The historical context of Remak's work is inextricably linked to the challenges he faced as a Jewish scientist in 19th century Germany. Despite his groundbreaking discoveries, he encountered significant obstacles in his career, including limited access to academic positions and a lack of recognition from the scientific establishment. The prevailing anti-Semitism of the time undoubtedly contributed to the delayed and often incomplete acknowledgement of his contributions. His story serves as a reminder of the importance of promoting diversity and inclusion in science and ensuring that all scientists have the opportunity to contribute to their full potential, irrespective of their background or beliefs. A particularly poignant aspect of Remak's story is his complex relationship with Rudolf Virchow. While Virchow ultimately embraced the cell theory, he initially resisted Remak's ideas and, crucially, failed to properly credit Remak for his crucial contributions to the Omnis cellular principle. This omission, whether intentional or unintentional, had a significant impact on Remak's career and legacy. It highlights the importance of intellectual honesty and the ethical responsibility of scientists to acknowledge the work of others. Despite this lack of proper attribution, Remak's influence on Virchow is undeniable, and his ideas ultimately played a pivotal role in shaping Virchow's own contributions to cellular pathology and his popularization of cell theory. The enduring relevance of Remak's work lies in his unwavering commitment to rigorous observation and detailed analysis. He meticulously documented his findings, carefully considered alternative explanations, and sought to integrate his observations into a coherent theoretical framework. His approach to scientific inquiry serves as a model for scientists today, emphasizing the importance of careful experimentation, critical thinking, and intellectual honesty. His emphasis on detailed observation and analytical rigor remains a cornerstone of scientific methodology, reminding researchers of the value of meticulousness in the pursuit of knowledge. Remak's influence, often subtle yet undeniably profound, continues to resonate within the scientific community. His story underscores the vital importance of recognizing scientific contributions, irrespective of social or political barriers, and celebrates the enduring power of scientific curiosity and dedication in the face of adversity. The legacy of Robert Remak endures, a testament to the transformative power of scientific inquiry and its capacity to advance human understanding of the world.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.